Well, hello. Thank you for joining us for another edition of Salmon Matter. This is a co-production of the Pacific Salmon Foundation and Conversations That Matter, filmed here at the Oh Boy Studios in Vancouver. Today we're going to be talking about salmon health with one of my colleagues from the Pacific Salmon Foundation, Dr. Andrew Bateman. 8 years ago, the Pacific Salmon Foundation embarked on a partnership with Fisheries and Oceans Canada and Genome BC to better understand how infectious agents were affecting the health of wild Pacific salmon. Naturally, this touched on the contentious topic of salmon farming, and of course, most salmon farming here in British Columbia is farming of Atlantic salmon in open net pens in our coastal waters, and the disease risk that might be posed to wild Pacific salmon from those farming operations. There was general suspicion that open net pen aquaculture operations were contributing to Pacific salmon declines, but the issue lacked scientific clarity. Research from that partnership that I mentioned between PSF, Fisheries and Oceans Canada, and Genome BC has confirmed that open net pen salmon farms do indeed pose a risk due to the transfer of parasites and other disease-causing pathogens between wild and farmed salmon. The findings prompted PSF to make a clear position that we believe that open net pen salmon farming in our waters needed to transition away from operating in the water where wild salmon and hatchery stocks of Pacific salmon could be impacted from a health perspective. Since then, our findings have informed decisions to phase out open net pen operations in the Discovery Islands and the Broughton Archipelago, both major migration routes for Pacific salmon. And in 2020, the federal government announced their commitment to transition away from open net pen facilities by 2025, a commitment that was again reflected recently in the federal budget that was announced. So joining me today is my colleague from the Pacific Salmon Foundation, Dr. Andrew Bateman, who is the Salmon Health Manager for our PSF Marine Science Program. Andrew, welcome to Salmon Matter.
Thanks, Mike. Hi, nice to be here today.
Well, it's good to see you, and I hope to actually see you in person sometime soon after we're through COVID. I know you've been busy. You also have a new baby there in the house, as well as your busy research agenda. Why don't you start things off by telling us a little bit about yourself personally, your background, education, what brought you to the kind of work that you're doing with us at the Pacific Salmon Foundation?
Sure. Well, I grew up in Victoria, actually, and I did an undergraduate degree at the University of Victoria, where I focused on math and biology. So I'm sort of a mathematical biologist. After my time in Victoria, I went and did a PhD actually in the UK, and that was studying meerkats. So long way from salmon. They live in the deserts of South Africa, in the Kalahari. But the whole time I was in England doing my PhD, I kind of couldn't wait to get back to the trees and ocean and salmon. So for the last decade, really, since I finished my PhD, I've been working largely on salmon and other marine species, dabbled in killer whales, and that brought me to PSF. And so I've been working with PSF for coming on 3 years now. Okay,
great, and we're delighted to have you. You've been a part of this research, as you said, for a few years, working very closely with Dr. Christy Miller-Saunders at the DFO, or Fisheries and Oceans Canada laboratories there in Nanaimo. Why don't we start at the very basic level of the definition of what does it mean when we talk about salmon health, and how does it play into, how does this research that you're doing around salmon health play into the overall context for how our Pacific salmon are doing? And I realize that anytime you ask how are our Pacific salmon doing, as scientists, this is a very difficult thing because it's a very complex web, very difficult to be generic, but do the best you can.
Sure. Yeah, as you mentioned, how are salmon doing is a complicated question. Many populations are not doing well, as many of us know. The same is true of salmon health, really. It's a bit of a complex question. Some people would define health in general as being free of illness or injury. But that's pretty narrow definition, and really the idea of health needs to touch on multiple aspects of an individual's life, whether that's a salmon or whether that's a person or any other animal. And so really it needs to bring into consideration the environment in which in which salmon live, the challenges we face day to day, as well as infectious agents and possible injury, predation, that kind of thing. Not for humans, we're not getting predated. But so really we're trying to see what role health of the individuals and the populations play in the salmon declines that make the media from day to day. And for the Strategic Salmon Health Initiative in particular, we were really focused on infectious agents, so bacteria, viruses, and whether they might be causing disease, a state of ill health in salmon. Because there are viruses that even as people we can have a cold and we might not say we're diseased. A bit of a stuffy nose isn't necessarily a big deal, and some infectious agents might not cause any detriment to a human salmon. So the question of infection and disease is a bit nuanced sometimes. So we want to see, are those infectious agents playing a role in salmon declines? Obviously, as you mentioned in your introduction, there's a potential role that salmon farming operations are playing. Salmon farms are a large aggregation of salmon that aren't naturally in our coastal waters. And so they present an opportunity for the farm salmon to pick up disease, whether that's naturally occurring in the background of the marine environment anyway. But the real issue is that they may amplify that disease. It may spread on the farms, become amplified above background levels, natural levels, and then transfer to wild salmon. And that may then cause a problem for wild salmon. Within the Strategic Salmon Health Initiative, we actually went further than just a focus on disease. And we'll get to that when we probably talk about some of the technology that's been developed within the program. But we tried to scratch the surface more than that, get deeper into some elements of other challenges that salmon face. And that may be high water temperatures or low oxygen conditions, things like that. Those all feature into the idea of salmon health. Because it's this multifaceted idea, you may be infected with disease one day, or sorry, not disease. You may be infected with an infectious agent one day and not suffer disease. But another day, if the water's too hot, or you've just gone through a very stressful life event, like a large scale migration up or down our coast, then that same infectious agent may pose a problem in that different scenario.
It's fascinating, Stefan. You describe it so clearly, Andrew. And I guess this is, you have to be careful about comparisons with human health, of course, but I feel like our literacy around understanding even what is a pathogen versus what is disease has grown immensely because of the coronavirus and the COVID situation that we're facing. And I guess, and you can correct me, the history major here, correct me on this, but we know that there's this notion of cumulative or compounding effects that happen in ecosystems in the environmental context. In other words, it's not any one thing that may necessarily be causing these declines in salmon, but certainly if we can hone in on understanding the various factors and certainly figure out the ones that humans can control in a more positive, proactive way, this is kind of where you're leading with this research in an applied way, correct?
Absolutely, yeah. Eventually, the goal is to take what we've found through this program, ongoing program within PSF, and be able to apply it as you say. So, like I mentioned, there are these multiple factors that might be influencing salmon, and absolutely they might compound. So it might not be that warm water or low oxygen or a migration or a disease would be overly detrimental on their own, but be that these multiple features together are what is causing big problems in some populations of salmon. And like you mentioned, there are some things we can control. There are some things that we have much less control over. A good example being climate change. Ultimately, humans are largely responsible for our ongoing climate woes, but we don't have an immediate way to control that within British Columbia or slightly more broadly year to year. So we have to take that as a given and really try to manage the system that salmon face by tweaking things that we do have control over. Aquaculture being one of those, pollution being another, agricultural practices, etc. is an obvious one. And so finding the balance and what the best things to do in combination of within those things that we can control for the best possible outcomes for salmon health and salmon more broadly is really the name of the game.
And there are parallels with human health, of course, too, in that the technology that scientists like you now have at your disposal can really significantly expand the knowledge of what is affecting health, the various factors that are out there. I often describe this project that we have, the Strategic Salmon Health Initiative, as being akin to the Human Genome Project. In a sense, we're using genomics, the ability to look very closely at genomic profiles within these fish to understand their health factors. Tell us more about technology and how genomics and genomic technology has advanced this type of work so rapidly. Sure.
I just want to say, when we're touching on the topics of human health and salmon disease, I want to mention that none of the infectious agents that we looked at or continue to look at pose a risk to human health per se. So they affect salmon or may be affecting salmon, but it's not like they're going to jump from salmon. If you go out and catch a fish on the weekend, you're not going to get infected by these salmon bacteria, viruses, etc. So that said, yeah, we definitely draw heavily on some of the technology that's been developed, as you mentioned, around the Human Genome Project and other ongoing work. And these techniques have become much more well developed over the last several years, decades. When I should say I'm not a geneticist, and lots of the technology we use does rely on genetic ideas, genomic ideas. So me, having done an undergraduate in biology, so I had some idea about the Human Genome Project and what that meant. I went away and did several years of ecological research, and then came back to work with the PSF and work with Christy Miller's lab at DFO. And I have to say, the technology that we now have at our disposal seems like magic. It's sort of wizardry of a scale that I certainly wasn't aware of. And some of this technology is pretty incredible. So I'll sketch out a few of the pieces that have become sort of our workhorses within the program, within the salmon health program. The key one that was the core element of the Strategic Salmon Health Initiative was a program to use, our technology to use genetic testing to screen for multiple different infectious agents within individual salmon. And it's really the technology that came out of the Human Genome Project that allowed us to do that at scale. So whereas the sort of old approach, the traditional approach would be to take a fish, take a sample of tissue from that fish, and if you wanted to say, is this agent here? Well, you might have to go and get a petri dish and swab the petri dish and culture the agent, see if you could find it. There's a bunch of other techniques you could use. If you wanted to use genetic techniques, you could run a genetic test for a single infectious agent. And if you wanted to test for multiple agents, you'd have to run test upon test upon test. Well, what the modern technology allows is what we would call high throughput approaches. So we can take a fish and we can screen it simultaneously for about 45 different infectious agents. And we can do that for about 100 fish all at the same time, which is pretty. So we have what we call these chips, and they go into a machine. They get loaded with fish samples. They get loaded with chemical reagents that allow the tests to occur. And on each one of these chips, we can run 10,000 approximately tests at a single go. And then we can use that information to ask questions about, okay, these fish are infected with one infectious agent or another. Now, how do we relate that to ecological setting, things we know about the fish when they were collected, et cetera? The other technology that we've developed as part of the program is the ability to take samples from those fish. So we, again, take fish tissue. And a traditional approach might be to take that tissue, slice it really thinly, put it on a microscope slide, and then look at it under a microscope. And you can do different things to illustrate different features of that tissue. So it might be a slice of liver, and you're looking to see if there's evidence of lesions, of signs of disease in that fish's liver. Well, with this new genetic technology, we can take that same sample and we can apply these kind of genetic tests, basically, and we can actually, rather than just looking at whether there's diseased tissue, whether there are signs of infection with a virus or a bacteria or certain parasites, we can actually visually identify the genes of the individual bacteria, parasites within the tissue. And we can see whether those genes of those infectious agents are co-localized in the same place as these potential lesions within the tissue. So it's a really powerful way to say, is there an infectious agent in this specific part of the fish? And is it tied to a sign of disease within that tissue? The third thing that the new genetic technology has allowed us to do is kind of go one step up. So we can say, are the infectious agents present? Are they affecting the fish tissue? And then we can ask questions of kind of the whole organism, the whole fish, more holistically, and look at patterns of what we call gene expression. So genes are the set of instructions to our cells. And the same is true of a human or a salmon. And when we face one challenge or another in the course of our lives, different sets of those genes may be turned on. Different switches are thrown within our cells that mean that the genes are turned on to different extents. We would say upregulated or downregulated within cells. And we can use this same high throughput technology to ask which are the genes within an individual fish that are turned on. And the thing we've done the most with that within the Strategic Salmon Health Initiative is to look at viral infections. So there are genes that are turned on within fish that are very clear indicators that the fish is infected with a virus. And so we can link up the infectious agent detection data with this gene activation information, and we can say it's infected with virus A, and it looks like it's suffering from infection. The really interesting thing we've been able to do is say, okay, this fish appears to be suffering from a viral infection or viral disease. We can't find any viruses in that fish. And then what we've been able to do then is dig down and use gene sequencing technology, again, leading on from the Human Genome Project. And we've actually sequenced the tissue in which we found the signal of that disease. And we've discovered brand new viruses that we didn't know about before using that approach. And so the SSHI and PSF have so far found 15 new viruses in just a few years using that approach. So it's pretty exciting scientifically. Potential big implications for salmon health and health and salmon populations generally.
You do a wonderful job of describing this, Andrew. Now, one question I was going to ask you, when you talk about fish for people who may be somewhat new to this topic, tell us what were did you just look at Pacific salmon? Did you look at just wild. Tell us what were the range of salmon that were included in this genomic study?
Well, there were a few different salmon species that we focused on, and for anyone who likes fishing for salmon or eating salmon in BC, you'll probably be aware that there you go, me too. You'll probably be aware that Chinook salmon, coho salmon, sockeye salmon are some of the kind of key species, and those are the focus on the Pacific salmon side. So from the wild salmon perspective, those were the three species that we mainly focused on and collected thousands and thousands of samples from various existing programs within DFO and other scientists collecting salmon samples up and down the coast. We looked at juvenile Chinook, coho, and sockeye. We also collected adult fish, so a really wide range across those species. And then, like she also looked directly at farmed salmon. And within BC, the majority of farms are Atlantic salmon, come from the Atlantic Ocean, aren't native to BC. We developed some partnerships with the salmon farming industry and managed to get samples of fish from salmon farms, Atlantic salmon from salmon farms. We also looked a little bit, not quite as in detail, but at the Chinook salmon on salmon farms as well. So we had information from lots of different sources, and that's really one of the things that led to the power of the program and continues to help inform the program as we move forward.
And there have been some very high-profile elements that have come out of this study. I wonder if you could talk about PRV, piscine orthovirus. See, I never get it right when I try to do it. You'll get it correct. But PRV, which was a pretty significant finding that we had a couple of years ago, and what we've found and what we're concerned about in that context.
Salmon farming and the issue of salmon health is a bit of a hot topic in BC right now, as many people will know. I think that boils down to the fact that people really care about salmon. Salmon are important for livelihoods, they're important culturally, they're important for ecology and the ecosystems around us. So it's controversial in many cases because there's just so much at stake for people. And PRV is a really good example of where the controversy seems to sort of boil at times. And it wasn't an initial target of the Strategic Salmon Health Initiative to focus on PRV in particular, but it had one of the infectious agents, one of the viruses that we've really spent a lot of time just because of the results that we started seeing. PRV is piscine orthoreovirus. Say that 10 times fast. And when it really came to the fore for the study was when a colleague of mine, Emiliano De Chico, who also works for PSF, started seeing evidence of the disease that PRV causes in Atlantic salmon. It's called heart and skeletal muscle inflammation. Started seeing that on fish in salmon farms in BC. And that was the first time there'd been identification of this disease. People, were pretty sure the virus was here, and we were detecting the virus within the program, but this was the first time that we'd seen evidence that it might be causing a problem. That itself has been quite a controversial finding. The program then went on to find evidence for PRV causing disease in Chinook salmon as well, Chinook salmon on farms, which of course raises the question, is it causing disease for wild Chinook salmon, not on farms? Because as much as we may be interested in whether PRV is causing disease for Atlantic salmon, that's not really our main concern here in British Columbia when we're thinking about the health of wild salmon populations. So it's like I was talking about before, it may be that Atlantic salmon farms are serving as a sort of incubator for PRV. It's infecting fish on Atlantic farms. And then the real risk we're concerned about is whether that PRV is transferring to wild salmon and causing them issues. There have been lots of questions floating around over the years about PRV. Where did it come from? When did it arrive? Does it transmit between farmed and wild salmon? Is it just naturally transmitting in wild salmon populations? And a recent study, what sort of our most recent study that's coming out in the next couple of weeks, led by Gideon Mordecai at UBC, uses again some of these fancy new genetic techniques, and actually some of the same techniques that have been used to study the evolution and transmission of variants of the coronavirus around the world. So using some of those same techniques has looked at PRV within salmon in BC and actually around the world. And what he's been able to show is that PRV came from the Atlantic Ocean. It's actually arrived in BC different times. It's quite clear from the evidence that it's arrived a couple different. It's also transferred from the Atlantic Ocean down to Chile. There's been a transmission event from BC down to Chile. So we can really track where the virus is moving around the world. In the course of tracking its movements around the world, we're able to say that PRV arrived in BC sometime around when salmon farming started. So it's not direct evidence that it was brought initially with salmon farming, but it's a pretty good indication. And then we can further dig down and identify patterns of transmission between salmon farms and wild salmon. And we actually see for Chinook salmon at relevant times of year, Chinook salmon that we catch in the ocean and then sample back in the lab are more likely to have PRV infecting them if they were caught close to a salmon farm. And so that's another line of evidence that really there's something going on here. Salmon farms are transmitting PRV to our wild fish.
Yeah.
And the final piece that we see from that study is that over the last few decades, PRV numbers, PRV infections throughout the Eastern Pacific around us have really increased over time. So it's not as if there's just some background level of PRV floating around infecting the odd wild fish, farm fish. It's really shot up in recent years. So this is one of the examples of the kind of thing that's possible when we apply these new tools to look at a specific agent. And there are a number of different infectious agents that we're looking at and trying to paint a picture of what's going on and whether individual agents and the suite of agents may be affecting wild salmon.
Yeah, and of course, one of the challenges that we have with this research, Andrew, is that, fish are in that black box of the marine environment, the Strait of Georgia, they're underwater. So we don't really have the occasion to watch how these things affect the fish necessarily and monitor that or see when they die, how they die. Maybe talk just a little bit about that, because my sense is that that's kind of at the heart of the scientific debate, because this is very contentious debate. We at the Salmon Foundation, based on your good work, believe we have enough evidence to think, let's err on the side of caution here, given how much our wild stocks are struggling. They go right by these farms. It's a tight, narrow corridor up there through the Johnstone Strait. So let's err on the side of caution and figure out a better way to do this, get these out of the water. But there are other points of view and credible scientists who have a different point of view. And back to the sort of premise of my question there, I guess it comes down to this question of, well, if we can't see that it actually causes something to happen, then we don't know that it's true. Can you just talk a bit about that sort of dichotomy that you're facing within the scientific community?
Yeah, it is a pretty complicated scenario, like we've said. And as you mentioned, there are different points of view, and there are different points of view about the science and also about what we should be prioritizing, as a province, as a country. And obviously from the Pacific Salmon Foundation's perspective, really wild salmon are at the core of what we do. So we're viewing things through a precautionary lens for wild salmon. So if there's uncertainty on a topic, we're going to say err on the side of wild salmon. There are clearly decisions we can make that that will be better at protecting, more likely to protect wild salmon populations.
Yeah.
You mentioned the difficulty in observing fish in the ocean. Yeah, it's sometimes described as a black box, as you say.
Yeah.
Fish go in, they're there for a while, and then we hope they come back. We hope we're able to catch them in the ocean or see them when they're responding to the river, returning to the rivers to spawn. And that's really one of the challenges of studying fish health in wild salmon, wild fish in general. If fish are on a farm or in a hatchery, even we can observe them on a daily basis sometimes, or even more. I mean, we can watch them hour to hour, minute to minute, and we can see if those fish might be developing infections, developing signs of disease, maybe not swimming quite right, or something like that. Whereas with wild fish, we only interact with a certain part. And so what we did within the Strategic Salmon Health Initiative, and what the information we continue to use is from multiple programs, like I said, that catch fish throughout that life cycle all around BC, and try and tease apart what's going on. And so for fish on a salmon farm, say, disease might be manageable if you get a bacterial infection as a salmon, and the salmon farmers can treat that with antibiotics, then it might not affect you too much. Whereas for a wild salmon in that black of the ocean, a salmon that even has just a cold, so to speak, might be a little bit worse at swimming. It might be a little bit less able to find food. It might be a little bit less able to avoid a predator. And so what are manageable infections or diseases in the context of salmon aquaculture may pose much bigger challenges for wild salmon. Yeah. So what we've done at that kind of broad scale perspective, the coast-wide population level view of wild salmon, is tried to ask, okay, we see or we may not see one or another of these infectious agents, but are the levels of those infectious agents that we see in those wild populations related to the patterns of survival and return of spawners that we know from other studies? And what we see is there's a mixed bag. So some infectious agents seem to be absent, for one thing. Infectious agents are not tied to poor salmon returns or poor salmon survival. But for other infectious agents, we do see a link. And PRV is one of those. There are some other agents. One that I work on is Tenacibaculum maritimum. So it's a bacterial infection, bacterial agent that exists worldwide, actually, and infects a whole number of fish species. But what we've been able to identify recently is it looks like it has been infecting Fraser River sockeye as a result of salmon farming in the Discovery Islands. And we also see that it has some of these population level impacts or associations with population level effects for wild salmon up and down the coast.
Now, I have a rule: when you use a 10-cent term, I always ask you to break it down to a nickel term. When you say population level effects,
Sure.
Help the history major and those out there who may not understand what that is. What does that mean?
Sure. Yeah, I guess it's kind of a catch-all phrase, yeah, and a bit of a biologist phrase. Yeah. So when we think of salmon and we think of salmon health, of course, we can think of individual fish. So you can catch an individual fish in the ocean. It may be infected. It may be obviously diseased or not. But what we're really concerned about from a management and a conservation perspective is all of those individual fish together. And individual fish that come back to a specific river or river system to spawn, we usually think of those as a population. And one river may have a spawning population of Chinook, it may have a spawning population of pink salmon, coho salmon, etc. There's different species, different spawning areas, and these fish have differences that are important to people and to the ecology of the systems. And when we're talking about population level impacts, population level effects, what we're really worried about is whether those populations, those groups of spawning salmon within rivers or river systems, are healthy and able to sustain themselves. So if we see a decline year over year over year in the number of spawning salmon in some reach of the Fraser River or the Skeena or some other river system, that's cause for concern. And what we've been able to identify within the work at PSF is to say, in some cases, those declines year over year seem to be tied to infections with individual infectious agents.
Okay, very helpful, Andrew. And of course, we know that, take sockeye in the Fraser River, for example. We've had the last 2 years, literally historic low, poor returns of those fish. And, going back, you were talking about the life cycle earlier. These salmon are so amazing and resilient. They have to go from the interior of BC through highly developed lower mainland, through the Strait of Georgia, up by the farms where we are hearing about these disease issues. And that's just kind of the beginning. They have to then go out to the open ocean, and depending on the species, can spend a couple of years out there. And we're hearing more and more from researchers about the challenges that are going on in the North Pacific, where the salmon are mixing together not enough food and warming waters. I just say all that to come back to that larger life cycle context, that when we do have sockeye, chinook, coho, particularly the thing about the Fraser River, that are many of those stocks are in such difficult shape. This type of research to help pinpoint those things that we can take action on sooner rather than later to try to improve those returns. So I guess try to have a question out of this for you. We have seen some policy changes here that have been significant. The federal government has said we are going to transition pens out of the water by 2025. The provincial government has said that by next year, about this time next year, that they will have new standards for issuing tenures or rights to operate farms in our coastal waters, which will be based on salmon health, the precautionary approach essentially, making sure they don't do harm to salmon, and also the rights of First Nations to exert their authority and rights on decisions. And this, of course, is another area of contention. Many First Nations want the pens out, and then there are some nations who have relationships with the aquaculture industry and coastal communities that depend on the jobs. It's very complicated. We look after the wild salmon, that's our focus, and the health, and we've taken our position because of the science. But my question to you is, long wind up here, I'm sorry, Andrew. The question is from a policy perspective and informing the people that make decisions about fisheries and salmon and whether it be in Fisheries and Oceans Canada or First Nations or the British Columbia government. How do you see this type of research that you're doing improving and informing those types of decisions? And are you optimistic that people are embracing this type of new technology?
Well, again, it's a complicated picture. Obviously, from PSF's perspective, as you say, we're focused on wild salmon, and from the fish health angle, we're really trying to do the best science that we can, provide the best evidence about how various stressors that affect fish health, salmon farms, but also, as I said, temperate oceans, other impacts fish may face, how those are affecting the fish that we love and rely on. But at the end of the day, it's not up to us to make those decisions about policy and directions economically and all these different things that as a nation, Canada, or as individual First Nations, we may need to make decisions about. So we can provide the best evidence we can, and that evidence really hopefully forms one part of the decision-making process that others in governments really, at the end of the day, have to make. moving forward, we're optimistic that what we've been finding and what we continue to find within the salmon health program at PSF and with our collaborators, Christy Miller, especially at DFO, but lots of others up and down the coast. We really hope that what we find can be used in a policy context, is relevant. We think it's relevant. There's some really exciting new tools that we've been developing over the years. Again, this is in collaboration with Christy Miller's lab at DFO. Really, she's the geneticist, genomicist engine that drives lots of this. And so, like I mentioned about the technology that allows us to detect, a viral disease in a state of viral disease in an individual salmon. Well, we can also use that same technology to look at which genetic switches are being switched on in a fish when that fish is too warm, or doesn't have enough oxygen, or is about to die even. And so we can use this new toolkit, which we call the fit chip technology, and we can use this toolkit at our disposal to start asking more questions about these multiple sources of stress that salmon face throughout the course of their lives. And it allows us to come back to that question of cumulative effects. What is affecting fish? And are the different stressors that fish face in combination having an outsized impact? And then we can ask, well, we know there's all these different stressors out here. We can assess the pattern of stress up and down the coast or throughout the life cycle of salmon. And we can then ask, okay, what are the stressors that we could best remove or mediate for these fish? What could we do to give them the best chance of exerting that resilience that you mentioned that we know is inherent to salmon? And if we can help inform policy through science by providing answers to those questions, we really feel that we've done our job. And that's the focus of where we're going to go over the next, and hopefully can be seen as the focus of what we've done over several years with all of this work.
Yeah. And kudos to your team too. You mentioned working with individual Indigenous and First Nations. We're invited by the First Nations in the Broughton Archipelago agreement to assist them with the scientific monitoring and technology side. And we have really, I think, to your credit and our Christy Miller's credit, Dr. Brian Riddle, our team with Genome BC DFO, I think that we've advanced this knowledge quite a bit. And I'm very keen to see where it goes in the future, particularly in the context of climate change. When you talk about the fit chip that you just shared, the kind of the future of the program, to be able to look at a variety of other stressors that we think are there, but we may not know much about. Andrew, before we close up here, I really appreciate your time. Anything that I didn't ask that you'd like to share with folks that are watching Salmon Matter? Any other topics you want to talk about?
I guess just touching on what you said there, that's actually a good point. Something that's really, really exciting and it's kind of an affirmation of the work we've been doing is that, whether it's First Nations or other groups that we work with, or even that we haven't partnered with in the past, there's much interest in adopting the technologies that have been developed in part by PSF in partnership with those other researchers, and using them more broadly, and not just for salmon, but for other species as well. But we're obviously focused on salmon, and so taking that technology and applying it in different areas of the province and even around the world. There's groups in Norway, for example, that are adopting the technology that we've helped develop. And so it's really exciting to see that. We think that really indicates that we're onto something here, and hopefully it can be useful from the perspective of wild salmon.
Well, thanks, Andrew. You are doing remarkable work, and you've spent the better part of an hour here really helping people understand the complexity, but doing it in a very easy to understand and succinct way. So thanks for all your work. I'm thrilled you're part of our team at PSF, and look forward to what you're going to come up with in the years to come. So thanks, Dr. Andrew Bateman, who is the Salmon Health Manager at the Pacific Salmon Foundation, for joining us. Thanks, Mike. Pleasure to be here. Thanks, Andrew. Well, and that will wrap up this edition of Salmon Matter on salmon health. We will have more to say about this because this is a topic that we at Pacific Salmon Foundation are going to continue to invest in with our partners like Fisheries and Oceans Canada, Genome BC. Have to add that our model is a partnership model, and we have many donors who contributed funds to this research and continue to fund this research. Too many to name at once, but we thank you, all the donors, and certainly partners at Fisheries and Oceans Canada and Genome BC, and the many First Nations now that we're working with on this research as well. So much more to come. Thank you very much for joining this edition of Salmon Matter, a co-production of the Pacific Salmon Foundation and Conversations That Matter. Keep well.